I'm honored to pass the stage to Dr. José Padron, full professor and head of the Biolab at University La Laguna, Tenerife. He leads the research group Biolab at the University Institute Bioorganica Antonio Gonzalez. His research focuses on anti-cancer drugs, with a particular interest in hard-to-treat cancers, with the ultimate goal to identify the biological targets at which the active compounds are directed.
Thank you very much for your kind introduction. Today I'm going to talk to you about two recent papers we have published using CXA as one of the tools that help us to follow the effects of compounds in cancer cells, which is our main tool of research. In the first paper, we described a method to prioritize the plants that we would like to study in a group in terms of the potential. In the slides, you can see that we define a term which is called the bioactive potential, and this can be calculated using three important and relevant parameters. One is extraction yield, which is related to the amount of compounds that dry mass that can be obtained from the plant using the solvent of preference or a mix of solvents. The bioactivity, which in our case is the anti-cancer activity, anti-proliferative activity, but you can use the one of your interest. And the chemical novelty, which accounts for the amount of new compounds that is not described in literature before for these plants or even for other plants that can give us an idea of how relevant will be to select these plants from the group of plants. In our case, we study 4 different Hypericon species growing in the Canary Islands. They are endemic from our place. Hypericon is distributed worldwide, and we are now studying how to compare our results with other plants. And we have seen that from these 4 species, applying the formula, Hypericon grandifolium appears as the most promising plant for finding new drugs. After this identification of Hypericon grandifolium, we decided to perform some tests on possible modes of action of those microextracts. In particular, we use the ethanol water microextract to analyze, and after some set of biological assays, we arrived to the live cell continuous imaging. And for this experiment, we treated HeLa cells, and we compared the results with HeLa cells treated with tamoxifen. We use tamoxifen because it is a drug that induces autophagy, and that's a characteristic we have seen before in the previous experiments. However, using the CXA, we were able to observe that the effect of the extract when compared to tamoxifen was completely different. As you can see in the videos, tamoxifen induces some effects on the cell, but it's not able to induce cell death, while our extract is producing cell death. It can be best viewed in the graph on the right side, where after 9 hours of exposure, we see that cell death speeds up. As you can see from the video, this cell death is mainly occurring by apoptosis. We, at this time of the publication, didn't have the LCD assay, the live cell death assay from an animal life, but we ran it later on, and we confirmed that these cells are dying by apoptosis. Another interesting feature that we can use CXA to study these plants, at least in a preliminary fashion, is to observe all the morphological changes that happens to cells until they are dying. And we can observe the whole population and see that cells die at different time points. There are cells that get vacuolized and looks more resistant, and there are cells that do not vacuolize and they are more sensitive. So this is open a new way to study how compounds, or in this case, mixture of compounds, can interact with these cancer cells. So it's very interesting to see that at least there is a difference in cell death between subpopulations within our sample. The second paper, which is one of the most intriguing part in drug discovery programs, and has been the holy grail in our group for the last 20 years, is how to find the mode of action, the mechanism of action of a compound that we study for the first time. So there is no precedent in terms of biological activity. And we believe that CXA is going to help us to achieve this together with artificial intelligence. In this paper, we publish our preliminary findings using NanoLife CXA and the phenotypic metrics that if software provided. And these metrics were transformed with a mathematical function, which is called the fast Fourier transform, into a series of data that was then submitted for artificial intelligence to cluster the results. In detail, we can say that we select drugs to test microtubule interacting agents, in particular Taxol, which is a stabilizer of microtubules, and vinblastine and colchicine, which are depolymerizing agents. Here we show the pictures at different time points, but in the videos we can observe that there is similarity between vinblastine and colchicine, while Taxol produces different phenotypic effects on the cells. We wonder whether the system was able to distinguish this from an computational point of view. So what we did was to analyze the 13 parameters for each of the cells, and all these graphs, which are shown on the top right figure, were transformed, this time series were transformed with the fast Fourier function into two parameters, the phase amplitude and the amplitude, and the phase. This two data was submitted to artificial intelligence in order to cluster, and we can observe on the heat map at the right side of the downside of the screen that colchicine and vinblastine were clustered satisfactorily together, while Taxol did not have any possible clustering with the other. So these results, although preliminary, it brings to us confidence that with the application of the live cell-less assay that we were not able to apply at this time, we could make stronger predictions. And we are working now on applying this to different other compounds with different mechanisms of action, at least the reported ones in the literature. So I really anticipate that this tool can help researchers working in drug discovery, at least in the cancer side, to get a rough idea of the mechanism or mode of action of the new compounds. And let me just thank you for your attention, and obviously thank my team for all the help that they provide to me.